Well, good evening, ladies and gentlemen, or lady and gentleman. Brent, good to see you. Pucker Up Genetics and Robert Federiconi and Dave. Hey, Gina, Franco, and Dave, good to see you. How are you? Oh my goodness, another week has gone by already. You get old, and days just fly faster and faster. What is it? October's almost over already. October 22nd. And we're back. Don't know what's going to go on. Hey, Sips. I think you did mention that you got your little package in Buds. I think you got your package, but I think they're a little worse for their wear when they left. But I'm curious if the light stayed on that long for a couple of days. If you can let me know when you got the box, because I had the light on when it left. And those clones, those clone containers, and also I don't think I mentioned last week, but hope you're not too disappointed, Buds or Run Boy. Run Boy had some different varieties, but I was a little worried about shipping them in that container because it didn't seem real stable. So I didn't send those to Buds. Okay, light was lit, lights on. Okay, and it looks like it was. Maybe I should have packed a little root cube, maybe with some more tissue paper or something. I don't know. So the lights made it on, so it lasts a few days anyways. Anyways, I hope you have some success with that stuff. It's actually a very good plant. Very good genetics. I'm glad we spread it a little bit around the United States. That's good. No more available at this time. Let me drop a link. Maybe we'll get some kind of content this evening. Well, maybe, man, a lot of people watching. What's up with that? Hey, Aussie, good to see you, man. How you been? And Aussie, I had a great conversation with you. And if you can give me that, I don't know, an easy way to send you a little something, I think I would like to do that. Anyways, what's going on this evening, ladies and gentlemen? Any questions out there? Hey, good to see you, Vets Grow. There's Buds. There's going to bail me out. I'm going to show some good stuff here. Well, I can't hear you, Buds.
Can't hear me?
I can now.
All right. Well, I'm at 9 weeks. With Purple Punch.
Wow.
Starting to fatten up pretty good and real dense.
Now is that one
like candy in here, real nice.
And that's one plant.
Oh yeah.
One plant, ladies and gentlemen.
3x3.
A 3x3 tent.
Yeah.
One plant with
fast lights.
Not bad.
Woohoo! And then there's the white goji. She's starting to fall over.
Those are some nice. Wow. Those look pretty good too.
Yeah, starting to fade a little, so they'll come down sometime this week. I'll harvest them sometime here. And then here's the little clone.
Oh, there it is. Okay, I thought that fan is really bending it.
Yeah, she came back. I mean, it was really limp when it came here, and the insides kind of flopped all around, But I didn't think she was going to make it, but I put the steamer thing on her and just transplanted her and steamed the heck out of her. And, yeah, she came back.
No, I think she'll be fine. Yeah, I don't think any of them made it unscathed. I think they were all a little roughed up.
I was glad to see her, perk up, though, after the next day I looked in, and she was all lifted her leaves up. I cut them off just to try and give her more opportunity to grow up instead of those leaves.
Well, that's good.
Green bicycles.
Look at that. Look at that's a classic shirt there. I
Oh
think
yeah,
I like that design. I think I
there
like
you
that
go.
one. Yeah.
Very cool.
I need to make a new order. What else is new, buds? How you feeling?
Pretty good, yeah. Things are going good. Oh, just getting ready to harvest. That strawberry glue, that outdoor came down and it came out real nice. I think it could have went another week or two, but where I had it, the guy was getting kind of paranoid.
So it was in someone's backyard.
Yeah, yeah. And he had stuff going on, so there was going to be contractors and shit there. And he got nervous,
Yeah. It's,
but it's all right. It came out good. It's not as couch locky as I like it, but
Tastes
still
good.
Yeah, it tastes real good. Has very sticky, nice smell. Yeah, it came out all right. He did bud wash and all that to it too, so being outdoor, he did a good job. I was proud of him. He handled it well.
Yeah, a lot of the outdoor stuff. It's all we got a good rain, or not a good rain, but we got a good rain. We got a rain yesterday, so yeah, it's all over now. It's all over
Yeah,
but the mold. So
yeah, that's what he was kind of worried about too, because we were getting frost came in and he had to cover it one night, and then he was worried that it was going to get frost and rain all in the same, and I think it was just, it was the first plant that he cared for outside, that got gigantic like that. Green bicycles makes them grow.
was he using green bicycles too?
Yes, sir.
I like that. And by the way, those plants that you showed, there's some green bicycles in there.
That's all
Yes.
green. I don't even add nothing else. Green bicycles. Oh, and a little Cal Mag from Humboldt, but that's it.
How we doing, vets? It's a big iced coffee there.
How's it going, gentlemen?
Wow.
What's happening there, buds?
Is that an
Good,
old
good.
fashioned chocolate milk?
No, it's
Coffee.
old-fashioned coffee. Yeah,
Oh,
I drink coffee all day. It's
I
like,
knew
no.
it.
I think, the third pot today.
Oh, you got a
Okay.
good signal tonight, Bets. You're not at home?
No, I'm at home, brother. I'm at home.
Damn.
And I did get my Blue Dream clone as well. It made it through pretty good. I wouldn't say unscathed. The couple leaves got caught in the little screwy part, but other than that, it was a little curled because it was laying down,
Yeah, mine too.
but yeah,
perked
she
right
popped
up.
right back up.
Me too.
Yep.
Mine popped right up the next day. She was all praying, lift their leaves up, happy. So, but
Yep.
I did notice that now that's the one that was in flower, right, Pat? That you took clones off because she's kind of in flower.
Actually, this is one. Those were from courtesy of Run Boy. Mine were even worse.
Oh,
So,
okay.
so yeah, I think that when I saw them, they looked like there was a. So yeah, you're gonna have to baby them a little bit, but hey, the genetics are worth it. I swear. You're going when you grow this one out, you're going to say, "Wow, this is nice."
Sweet.
I hope.
No, yeah, it'll dream badge, no problem. It was
I
still
think so.
small. Yeah.
If not, I could always get a new one and we can redo this little experiment. But yeah, I'm glad they shipped okay. Who Runboy was saying there's better ways to do it. He saw a little container, wasn't real impressed. I didn't want to put 3 plants in that for maybe that would have made them more stable. I don't know, but he had a couple extra varieties for you that you didn't get, especially for you, buds.
Well, it's like the box was smashed, like really smashed on one side. So I was worried that it had popped open and everything else, but it was still intact, screwed shut. It's just that it just kind of floated all around in there.
Yeah, I probably should have said handle with care, but I don't know how much that matters.
Yeah, no, it made it fine, and like
It
the
made
light
it.
was still going, and it perked right back up. I slapped it in soil right away. I didn't know So yeah, I just put it in some soil and yeah, she perked up.
Yep. Well, good.
Very cool.
I spread some genes.
Yep,
Not my own, but hey.
that'll be a spring harvest over here. Add to the
Yes.
collection. I got some grapes growing. I got a couple more caramel creams growing. I got the sour apples growing. So we're gonna have some fun.
Oh, that's good. That's good.
You
Oh,
know,
it's
back
really,
in the day, we used to just get a wet napkin, get it wet, and put it inside of a plastic bag. Put your clone end in it. Put it in a CD case, and put it right in the mail.
Yep.
Allegedly.
Yeah, allegedly.
we've got the thunder from down under up on there. How you
Oh,
doing?
hello.
What
How
up,
are you
Mike?
today?
Hey, what's happening, Ozzy?
How's it going, guys? Join you for a sesh.
Good. How's the weather there? It's getting to be summertime.
In the northern part of Australia, yeah, but I'm in the southern bit, so it's still transitioning, I suppose is a good word. Yeah, it's all right. Thanks.
I stopped into your live show. I think it was yesterday. Very impressive.
yeah.
If you're not in Aussie CC, he's got a YouTube channel under that name. Definitely check. Definitely some information worth looking at.
Thank you. I try and structure some shows to have titles, and then I can focus on different topics. It's good. I sort of do them a couple of weeks in advance, but it's hard now because my uni's just finished. So I used to pull stuff out of my uni and think, wow, this is going to be rad to talk about. This is so related to cannabis. But it's finished now till I start up at the end of January. So, yeah, we'll see how it goes. Over.
I'm gonna jump back into chat because I can hear better in chat. So.
Hey, Buds, thanks for showing us some good content, sir.
Oh,
Good
yeah.
weed, mate.
Thanks
You're
to
doing
you.
well, buds.
So, anything new in your sphere, Aussie?
I suppose I can crack one. It's not really going to help you guys, but I crossed a couple, and one of the outcomes was amazing. I've had this triploid for a while, and I haven't done it through cytotomy. I haven't diagnosed it that way because I don't have that meter, unfortunately, or that electronic device. But you can do it through stomatal size, and through looking at your triploids and your tetraploids, and it goes on your size of stomata of the size of them and their area per square inch. So if there's a triploid or a polyploid, usually they've got massive stomata and the size per square inch is a hell of a lot lower. So if you do I've done quite a few leaf samples and put them in the square and then count them all up and then write it down and put it on a chart and all that sort of stuff just to prove that what it is, I suppose. And I've done it with normal ones, with diploids, and you can see there's the difference in it. But anyway, and also this one, I couldn't herm it out. I really tried. I tried. And finally, after a few years, I've had this for years, and I couldn't do anything. I tried to stress it, put it in front of the fans, everything, nothing. But I ended up pollinating it successfully. And I did it with my keeper, of one of my keepers, Pink Death Bubba, number 2. And it came out massive seeds, the biggest I've ever seen, and which is a trait of ploidy. And the terp profile's off the head. It's I have to say it's at least 6%. I know it's really high, but I reckon it's at least 6. It's my normal stuff's usually about 3, and this thing is just so loud. It's really nice. So that's something new, I suppose. Yeah.
So give me some of the terpenes in there. You think?
It's a sweet rubber comes to mind. And it's like a very chemically induced, like you're walking into a lab where someone's mixed up a lot of stuff. But it's a sweet industrial sort of like rubbery smell. It's unique. It's not familiar to me, and it's not familiar out of the parents. I can't pick either of the parents out of it, which Lavender Bomb and Pink Death Bubba. I can't pick any of their lineages in this terpene profile mix. It's great.
So what was the time frame for finishing on that one? Indica dominant or sativa dominant? Remember?
It's definitely indica dominant. All the stuff that I do is all indica dominant because I got caffeine sensitivity. So sativas, I ping off them. Can't sit down. It's off them type of thing. So it's an indica dominant. So it's the first run, and I had a problem with this run. So believe it or not, can I say that? I don't like talking about my stuff too much, but I just had a problem with this run, and it wasn't very good. Pseudomonas syringae got entangled in the tissue, and it created chlorosis, and it was very hard. So yeah, it's hard, so I can't give an accurate answer on this to when it's finished.
What does it look like? I'm a little curious.
Shit, it's nothing special.
Oh, I wish I could
I wish it was not. It was a tester, so I only put a little one out, and it went. So it was just to see what it was like, because it wasn't growing too well in veg. It just wasn't. I thought it had some mutation of some type, but I don't know if it was the conditions. I only did one tester, so you can't really grab much out of one. But yeah, that's sort of new.
so was there only now you grew that from seed, right?
Yeah.
And did you only grow one plant?
Yeah.
Okay, so we don't know what could be next time, right?
I've still got I made about, I think, 20 seeds of that variety. So there's still some there. I just wanted to test it because it was unique. I couldn't believe that I'd actually pollinated it. It was fantastic. They reckon that 90% of triploids are infertile. So it's a good trait to have in your genetic line to prevent herms and stuff like that, so people can just thrash it out. And that's why I've been mucking around with tetraploids, inducing diploids to make tetraploids, and then you backbreed them, and then that produces your triploids, and that's what gives you your extremely stable lineage. Like what's those? There's a few cultivars that are renowned for that. They've got that triploid function feature in it, which gives it a hell of a lot of stability. Yeah, that'll do. I don't want to hoggle your airtime.
No, so would it be the opposite? So that would be something you probably wouldn't want to work with, say, autoflowers, or maybe would that work with because autoflowers, it's the opposite, right?
Yeah, or I have done a little bit of work. Well, I pulled a gene out of an autoflower, an ELF gene, because that's a predominant trait that makes them finish early in ruderalis. Mm-hmm. So I pulled that out and I've whacked it in. Pineapple Death Bubber, I think's got it, because it finishes on day 50. This is honest to God, I'm not making shit up. Day 50, I've got amber trikes and there's no more white pistils. And I've done this over multiple harvests, so there's nothing that's infected it, or it's no abiotic or abiotic stresses has caused it to finish early. Yeah, it's really cool. That's about my experience though with the autoflowers.
So
I'll show you some shit. Just
now you mentioned you pull a gene out of something. So are you doing this with like CRISPR?
No. Do it through the 3 ways of marker-assisted breeding. You can do it through morphogenic expression, through chemical, and with the chemical it would be like staining and stuff like that. And then the third one is your molecular, which is your CRISPR and your DNA technology with your you're getting in with your electron scanning and your PCRs and stuff like that. So I just do the first one with morphologic expressions, and there's a lot of things that you can do, traits in that which you can breed with, and the visible traits are, they're visible, so it makes it a hell of a lot easier. And the ELF gene is a visible trait, so it makes it really easy to see what's going on. You can see which is finishing, which isn't finishing, so it's pretty cool.
Okay, so was the seeds the I'm going to say it wrong, the polyplid, did that pass on?
They reckon that if you breed with a triploid, it's supposed to make a tetraploid, because the 3 chromosomes that are inside of it's supposed to not halve. The outcome's supposed to be a tetraploid, which is 4 sets of chromosomes. Yeah, That'll do.
Now, does that bear out in, because I always thought of that, there'd be like 3 leaves instead of 2 leaves.
No, that's a.
What is that?
I've had that one. What's that one called? That's a. What's the name of that gene mutation? Shit, I've got it written down somewhere because I've had it before.
So what's
It's a not a branching BRC branching gene mutation. No, because it's a cotilina branching gene mutation. It's not out of the nodes, so it's not a BRC. It's a cotyledon branching mutation, and they said that that's an advantage in the cultivar where it can start off a lot easier by putting 3 sets out instead of 2 sets, just to absorb this photosynthesis to start. Over.
So what were the characteristics of the triploid? That is there, and you said they're visual. How do you know you even have one?
The triploid, the characteristics. The stomata is the main thing.
Okay.
So if you get your compound microscope out and put it onto, I think under 20 I can see mine on, or 20 or 10, I can't remember what I put it on, and then you can zoom in, you can take photos of it, and you can do the area, and then I've taken, Jesus, I'll just show you.
Show me.
Triploid, where is it? Ploidy, there it is. Because I'm doing some experiments with some overseas people, some of university in the States that wants to find more information. Another fellow in university in Europe, Switzerland it is. Where's the present? Share screen. All right, I'll just give it all, stuff it. Ploidy. So, what do you want to know? What was your question again, please?
Like, what is the visual characteristics that you know you have a triploid?
The triploid versus a diploid. So up here, you can see over here on the left here is your diploid, and on this side is your triploid. So it's to do with the sets of chromosomes. So you can see here the size of the leaves. So look here on the leaves. So the leaves are massive, and it's the middle one. So the middle finger blade, see that one down here? It's just absolutely giant. That's the big, like your middle finger. That's a very good characteristic of it. And see here, they put out more leaf size area so they can grow more. And then see down the buds, it's hard to tell with the buds, but it's just more denser and more filled in. That's one characteristic. Here under the microscope, you can see the resin content. This is cultivar dependent though, but apparently you're supposed to get a little bit more of the sesquiterpene terpenes in triploids, in your polyploids, tetraploids, and polyploidy means more, and ploidy is the chromosomes. So poly is Latin for more, so it just means more sets of chromosomes. And then you break it down. Di means 2, triploids 3, tetra is 4. So that's how you get your chromosome count with these, what we're talking about. And they reckon that you're supposed to decrease your amount of THC in your tetraploids, and I think that's true. And it increases your CBD, and it definitely increases your terpene profile in sesquiterpene terpenes.
So is that the heavier terpenes?
Sesquiterpene is what's the isoprene? It starts off in your monos and then it goes sesquiterpene. So it's your second isoprene level with your 10-block carbon chain.
So those are a little less volatile.
Yeah, I think. Here's the difference in your diploid and your tetraploid leaf. So this is the 2 sets to 4 sets of. You can see the middle leaf is very, very prominent. Actually, no, that's not different. Down here, you can see definitely is a thing they use in the lab, and you can define it by your stomata. So see how there's a massive difference in that. See the scales on the bottom right-hand side here, this black line? And if you compare the left to the right, they're nearly double the size. I'm pulling out, these are probably about 15 to 18 micron. And over here, I'm pulling like 30 to 40 micron for stomata length. Massive difference. That's how to make it. Actually, better stop sharing all this stuff. I'll get back and I'll show you some other photos of some. What do you want to see?
So I'm wondering, when the stomata are that much double the size, do you have to approach them like foliar feeding differently, or any more or any less?
That possibly this is the stuff this is only new because it's not new to science, but it's new to experiments in cannabis. It's been happening in genetics for generations of years, and they said that this is a thing. Like, did you know tobacco is tetraploid, has 4 sets. Strawberry has 6 sets, which is hexaploid. There's in generations over the years, Mother Nature kicks in, and when events happen, they tend to the plant. There's multiple ways it can induce itself to double its chromosome set, and one of the ways is through genetics, through the environment. Is when it comes to like an ice age, for instance, it'll shit itself, and only a few varieties will try and reproduce, and all of a sudden, when they're doing their pairing of their cells to reproduce, their chromosomes they don't halve like normal, they'll double, and that's when you'll get your ploidy will happen. Here's some no.
So can they usually can they trace back like that change? Like if there's something that's tetraploidal has 4, is there a species that will have 3 before it or 2 before it?
Yeah, they've checked it back with evolution. Yes, you're right about that.
That'd be interesting.
Yeah, it is pretty topic.
So something like a UV event or something, some solar flare or something could. What else could cause the chromosomes to change that dramatically? I don't know.
Heavy, near occurrence to death. So something that's really, it's just struggling, it's just hanging on. How can we hang on? All right, we can try and induce, and we'll double our chromosome. Because with you've got 2 sets of chromosomes, you've got extra sets. They're not really doing much. Because on your chromosome leg, for instance, you've got 2. They've got all your genes on them. So a plant's got 35,000 odd genes, the cannabis plant. So on one set of chromosomes, you've got your genes on each leg. And then each gene can interact, and then it interacts from signals, and then it goes and processes in a signaling cascade pathway. And that's what gets you all your communication that happens through your chromosomes. So if you've got an extra set, they're not doing much. So they can take care of problems, diseases, stuff like that. So the ploidy plants tend to handle biotic and abiotic stresses a hell of a lot more. That's why triploid's a good feature where it's very hard to harm. 90% of them are infertile.
Interesting. And that came from a ruderalis, you think?
No, that was the ELF gene came from the ruderalis.
Oh, okay.
The early finishing gene, because that's okay. It finishes the period. The photo period finishes through its genetics, not through light, not through time. You know, like our normal indica or sativa, they'll get induced through light, and then because they're a long day plant, so they'll get induced through light, and then it'll flip. Where the ruderalis just go to the genetics, and then their genes tell them when to flip and when to finish.
Now that's all. Now I don't know how much. What's always fascinated me is what there's a clock molecule in a plant that measures sunlight, and it's counting. I mean, that almost has to happen, right? And that would be a real interesting molecule or process. I would find that fascinating. I'm sure it's complicated as hell, but there has to be a clock molecule inside at some level, right? That's measuring how long the sun's out there.
The plant works off the circadian rhythm, believe it or not.
Okay.
So you did right. There is a clock. And all the processes, if I had a leaf porometer, I could prove that to you too. And that's one of the things I'd love to get. $600 they are, and it measures stomatal conductance. I'll show. I've got a slide on this. It's so fascinating how the plant just functions. Like if you get your plant regular in a pattern for coming on at a certain time off at a certain time, it'll know that. So it'll go into its sleep phase at certain times, and regardless if you put the light on or off, it won't really affect it. It'll affect it a little bit because it'll start to alter it, but the plant knows because it's got a circadian rhythm. Yeah, I don't know how to explain it.
So somewhere that has to be a molecule that's a clock of some kind.
There's a few genes, that are related to circadian rhythm. I'll see if I can pull up the actual name of the genes. I don't know those ones, sorry. Photosynthesis, no. I've known it because I've talked about it a bit, so I know I do have some stuff on the plant circadian rhythm. I just don't know. Can't.
Do they do a daylight savings change in Australia?
Yes, it sucks.
Yeah, I don't know. I don't understand that.
Bloody pathetic, mate.
Stony Creek, thank you for stopping by. Ron, good to see you. Martial artist. Martial artist.
He's a cool fella. Knew him
You know,
quite a few years ago.
martial artist.
Yeah.
Oh. Welcome. Thank you for tuning in, Sips. Well, all those kind of molecules fascinate me, you know. And were you the one saying that THC might be produced on the outside of a plant?
No, it's produced it's a secondary metabolite, and secondary metabolites are stored in various parts of the plants, in the vacuoles and dermal tissue, in trichomes, in edioblasts. So there's a few different spots where things can be stored. That's why when you break up the plant tissue, you'll release other different volatile compounds, because they're stored inside the plant tissue as well.
So is there an understanding? I know from my understanding, you got photosynthesis, we create a sugar molecule, like carbon, oxygen, and hydrogen. And that molecule gets folded and added to, which eventually is carbon, hydrogen, oxygen. For CBD is THC, and all the terpenes are just carbon, hydrogen, and oxygen. And they must all start from that sugar, would be my guess.
Well, they start from CBG, is the mother in the cannabinoid family tree, it's called. And they've happened from their signaling pathway to initiate. I haven't got that one.
But that carbon, hydrogen, and oxygen must come from that sugar molecule, would be my guess, because that's that photosynthesis is pulling in the carbon, right?
How does it assimilate the carbon? It uses it through. You're getting into those Krebs cycles, and those cycles that are bloody hard. You know, that the glucose cycle and those things that does the actual, where you take 18 molecules of that and turn it into 6 molecules of that. I'm not real familiar with that, to be honest.
No.
I'd have to be pulling up charts. I can't pull it out of my head. Sorry.
Yeah, I just would, there's not a lot of easy ways, I imagine, to bring carbon into the, absorb it through a CO2, which plants do. And I'm thinking that sugar must be the starting point for all the hydrocarbons in the plant, right?
That's no sugars. It's energy source. So it produces the ATP, which is the currency of the cell. So it's adenine. The ATP, adenine thiophosphate. It starts off at ADP, and then they can go through the cycle, which you're talking about with the photosynthesis, and it changes into ADP, adenine diphosphate, to adenine triphosphate. Yeah, that's where it goes into, and that's the currency. Sorry?
No, that's why actually phosphorus is real important. It has nothing to do with THC. It has something to do with ATP. I mean, that's what you want, right? You need that. That's the energy source. But let's say the 5 carbon terpenes, right? Where's that carbon come from? I would think it would come from that sugar. I mean, the sugar must do a lot of things, because it's a basic building block of cellulose and everything, isn't it?
Not particularly. I'll show you I've got a fair bit of this, so let's pull up that slide.
All right.
On the secondary metabolite profile. So I'll get out of the ploy stuff. Study, plant science, secondary metabolites. And you wanted the pathway to the pathway to grow them. You wanted the chemical pathway for it to work. That was is this one? What's this? The monoterpenes pathway. Here you go. Here's one of them. This will give you a bit of you like all your chemistry. Which one is it? This one. So this is a monoterpenes pathway. So this is just with your isopropene level with your 5-carbon chain. So your pathway up here. So it goes from your leucoplast, and then it goes through the GPS with that phosphate into limonene to the endoplasmic reticulum inside the cell. Then it goes through into the mitochondria, and then into the cytosol, and then it goes through all these processes to produce all these different types of terpenes.
Wow. Where did you get this information?
Study like my friend. I got heaps of shit like that. I got a lot because we are, as a medical cannabis person, we farm terpenes and cannabinoids. That's it. So I've got a hell of a lot on secondary metabolites, which is cannabinoids and terpenes. That's, because that's all I'm interested in. So I've got all the pathways, how to make them, what initiates them, what genes to turn on, that sort of stuff. Salicylic acid, ethylene, your jasmonic acid, those ones that initiate them are very, very beneficial. They'll give you great responses. So I've got, under that, I just showed you one chart. I've got a lot. Another 30 charts to show you under those things.
So do you think you could influence like different terpene profiles within one cultivar? Could you figure out how to emphasize different that would be an interesting thing to mess around with.
I've done it through. I've messed around with it a fair bit because I've been doing this for a few years. With Pig Death Bubba, I can bring out 3 totally different terpene profiles out of the same mother plant
By using different inputs?
Different abiotic and biotic stresses. Yes.
Oh, okay.
That's the answer.
Okay.
Because that's how you initiate them.
Well, I actually had this conversation, and I believe this to be somewhat true. Someone mentioned about growing cocoa leaves, and I said, well, you can't grow them at sea level. I mean, you can grow them, but they're not psychoactive. They need to be stressed. If that, however high they're grown in the Andes, that's not easy to grow, and And I've always thought about cannabis. I always like to stress them a little bit, and I think they're a little more pungent that way. But yeah, I think psychoactive plants respond to stress in a good way.
The secondary metabolite profile is alkaloids are the second section. There's 3 sections of secondary metabolites. You've got your terpenes, then you've got your flavonoids and alkaloids, and then you've got your nitrogen affecting terpenes, which is a real one we don't really worry about. So it's really the first 2. And that's where the alkaloids is part of the same profile. So by what you're saying, I believe to be correct as well.
Yeah, and I just don't know about like what you've done. You seem to stress them differently to like you've done a little bit of exploring, like different kind of stresses release different kind of, never thought about going that deep into it, but that's awesome. Hey sir, you need to write a book one day.
Well, it also don't have to be the stresses either. Like we all just got these clones of this Blue Dream from the same mom. They're going to be in different environments. We're going to add our own little stress to them. And I guarantee if we all got together after we harvested, they'll all taste different. They'll all smell different.
That's because your genotype equals your phenotypical
expression.
Your genotype equals your phenotype plus the environment. Your phenotype equals genotype plus environment. That's why. So phenotype equals your genotype plus the environment. So that's why I've gone and done 3 different outcomes from the same mother plant from altering the environment, which is your abiotic and biotic stresses.
I would say though, maybe I'm wrong, but if 5 of us got together in a month, 6 months, and we all sat down with our Blue Dream that we grew from that same clone, we'd know it's Blue Dream.
Yes, it's similar, but it does alter. So yeah, you're right. If you do to the connoisseur, you'd be able to tell it, but to the non-connoisseur, they would just know it's all the same. Here's the interesting thing. I had my pain specialist. He asked me a question about one of his patients that she's gone. Why did her medicine change when she buys her CBD oil from the same supplier? And I said because of the secondary metabolite profile, because they changed the abiotic and biotic stresses. So if they were to get a profile printout of their last one and this grow, they would see the differences. And then they'll know, all right, well, she's not just working off CBD, she's getting entourage effects through different terpenes. So they could see which ones that worked last time with her, which ones are minus this time. That's the specific terpene that's interacting with her body. Over. Oh, Jesus. Good fun.
I'm always glad when you stop in, sir.
Yeah, we've still got to have our little chat too. Hopefully later on today, if you're not busy.
Yeah, yeah.
And you know what's cool too is like, since we all have this blue dream, we can all be like, okay, well, that one has this, you know, terpene profile, and it makes you feel this way, and this one has this terpene profile, and it's this way. But with what Ozzy is saying, like, we could be like, okay, well, we need more limonene or more, whatever, and we can, on the next clone set or batch, we can induce the stress that we need to put onto it for that expression to come out in that next batch.
Correct. I got a call. I better go. I'll speak to you later, Green Jeans. It's good chatting to you.
Take care. Thank you for stopping in, sir.
So everybody chat to you next time. Bye-bye.
Always good to see you. Oh, Sun grows back there. He's going. So the Cowboy Cup's coming up. All the shows are. This is the show season coming up now, isn't it?
Yeah, I believe it's like December 3rd or December 5th, whatever that weekend is, right around there.
Oh, okay. Yeah, I forgot. That's when the big one used to be out here, and I think it's changed completely. The Emerald Cup. That was always a fun one to look forward to going. But I don't even know if it's where and when it exists. It still exists, but anyhow.
So our guest, Mr. Ozzy there, that dude, I really like that guy. He hangs out over there on Humidor's show, Humidor and the Flavors or whatever. But yeah, he's I don't want to say he's a scientist, but in a way, he's his own kind of scientific chemist. He's got a lot of really great information. I love listening to him talk.
No, and he's passionate about it, and I can hang my hat on a few of the words he says. I could just bear enough to make it interesting, and I'm focused. Yeah, no, real interesting character. I think all of us are. No,
but that is a great point, because we can.
He is absolutely a scientist, for sure.
Yes.
No, he does. What he does is science. So he's 100%. He's a scientist. So. But
yeah, we can. When we actually get to a point where we're actually trying to make some real medicine out of what we do, that's the science. That's the new stuff to be studying, not this light, that light. No, this is what happens in the plant, why the plant reacts, and what the reaction does. The
organic chemistry is what it's all about with that.
Mm-hmm.
No, and it's going to be guys like him, and and Dr. White, and those are the guys that are going to blaze away. And I'm pretty sure, I don't know his whole background. Someone mentioned he's a PhD. I'm real sure I will guess if he doesn't have one, he's working on his. He's just he talks about really high level science. Actually, that's his vocabulary. So
right.
No, I think he's a very interesting young guy, and I would love to have him work with Green Bicycles. So you never know.
Absolutely.
You know what I'm saying?
Absolutely.
Because he's all about doing the right things, and he's knowledgeable. So you never know what could happen. You never know.
Well, that was great having him on. That was a good bit of info for us this evening.
A lot of it could probably get overwhelming, but no, because I think about some of this stuff. It's like, where do you find that information? Well, if you're in the learning environment, it's a little, you're exposed to a little, that's my excuse, and I'm sticking to it. But I guess if I dug out, but I love that the pathways of what he was talking about, the different terpenes. Like I'm real sure it starts with sugar. That's where we get the hydrocarbons from, and then I'm think that just gets sugar does a lot. I could be completely wrong, but I have a feeling that anyways, I'm fading myself.
Mhm. Well, those we know we've got the building blocks of what the carbons do and what carbons the cannabis plant needs. It's what he's kind of more referring to is like all the stress, whether it's environmental or chemical or the stress from like this year my plants got attacked by bugs. That plant stressed out really bad and it fell out about week 5. So
No, or just like in the chemical path, like what stress does something to cause a chemical reaction to happen. So knowing about those pathways, yeah, because I was going to ask him about what is it called, terpinator. A lot of people ask what's in terpinator, and you're not going to find out what's in terpinator. And people swear it works. I've never used it. I don't have any idea what's in it, but I'm just wondering, if it's hormones or something that are important for some of those pathways, and if you just throw those in because they're I don't know how you would look at about going it, but the more you know about that chemistry, even at the stress level, if you're stressing a plant, something happens to happen chemically that has to that had to influence a chemical reaction somehow, and that mechanism is
for
fascinating
example,
me.
well, let's think about it this way for a second. So we have the Green Bicycles compost tea. So this is a bacterial dominant tea. Well, there's thousands or millions, however many different kind of bacteria they are, and I'm sure that different types of bacteria are going to add a stress level to the root zone if you implicate different microbial species, right? But that would make sense, right? So there's different stresses. Those microbes are transferring nutrients different than, say your native compost tea. Yeah, I get what you're saying with stress and stuff, but even we could do some kind of test with the differences in compost tea.
No, and I think when you look at it, that the biochemistry in those, he was talking about the Krebs cycle, which I believe is a carbon cycle. I don't even remember, but anyways, that the chemistry involved is pretty complicated. As a matter, I don't know if you know this. Because I like to talk about this about energy, but like when we breathe in oxygen, we burn it with sugar. It's a combustion reaction, and we breathe out CO2 and water. It's the exact same thing that happens in a car engine. Happens in our body. We turn hydrocarbons, which is we use sugar. Gasoline is the hydrocarbons that cars use. It's the same thing. It's just carbon and hydrogen and oxygen. But our process chemically in a car, it happens instantaneously. So you get a lot of heat. But in ours, it might take thousands of steps, thousands of reactions for that same sugar to turn into heat. So, but it's fascinating, and I don't think a whole lot. I don't know how much is known. He would know, but those kinds of it's amazingly long and complex. Some of those chemical reactions in biochemistry is what I'm trying to say. And I'm fading. Like I said, I'm fading pretty fast. We've had a pretty good show.
Isn't it when you move one molecule to another? Certain molecules have binding elements of heat or cool and how quickly they move them. But when you move one molecule from another, that friction or that snapping point is what creates the heat. Isn't that what it is, or something like that?
What creates the heat? Well, friction usually creates heat, but I'm not sure the question. I'm a little, like I said, sir, I'm fading. It's kind of late. So,
All right.
anyways, I'm thinking about wrapping this up unless you got something important.
Nothing really. Was Halloween next weekend, or is that the weekend after?
Oh, 7 days would be the 27th, 29th. So I don't know if they do official. It looks like it's going to be like Monday.
Oh yeah, so we got one more show before Halloween. All right, so next weekend's the Halloween show. Are you dressing up next weekend?
I haven't thought about it, but maybe I will now.
Right on. Right
Something
on.
something I have never done, but hey, you just never know. Are you going to dress up?
I don't know. I may. You know what? Every time we do something, I do come out with something silly on or do something silly. So yeah, probably.
I should come out as troll clowns or something. My puppet. But anyway.
Yeah. Well, last time you said you wanted to talk about aliens, and I came out with my little hat
Oh,
and
that's
all
right.
that stuff.
That's right. I don't think that went over very well. I don't know. I think I better stick to I don't know too much about many things, and I know just enough about cannabis to get me in trouble. But having you guys and guys like Aussie stop in and Buds and show what's up out there and sips and puffs and sun grown and yeah, I've got actually I got some good panels. Got some pretty cool panels once in a while. Hey Deb G, good to see you.
Yeah, other than that, we got Halloween coming up next week, and then that's or the following whatever Monday or whatever it is. But yeah, no, nothing else for tonight. If you're ready to wrap it up, we can get out of here.
Yeah, I could. I got a few things I got to take care of this evening, but anyways, yeah, thanks for coming.
Well, thanks for Ozzy. Yeah, thanks for
you.
Ozzy showing up. The chat. Thanks everybody.
No, that's good. All right, well, take care. Have a good night. Get a good night's sleep. I know I will. I know I will tonight. And peace. Let's